प्रिंसी, ऑनलाइन वाले, वो तो फंक्शंस बोला है, स्ट्रक्चर भी तो होता है। प्रोटीन। गुड इवनिंग। अच्छा देखो, राइबोसोम जो है, these are made up of RNA and proteins. RNA का फुल फॉर्म क्या है? राइबोज न्यूक्लिक एसिड्स, and it is rRNA, ribosomal RNA and proteins. There are two types of ribosome: prokaryotic ribosomes and eukaryotic ribosomes. Prokaryotic क्या होता है? Prokaryotic, it is a primitive cell where most of the cell organelles are absent, and particularly those organisms which are bound by membrane. So what are the cytoplasmic organelles bound by membranes? Yesterday you have seen, जैसे nucleus is bound by membrane, Golgi body is bound by membrane, lysosomes bound by membrane, mitochondria bound by membrane. They all will be absent in prokaryotes. ठीक है? So in prokaryotes there is a kind of ribosome. In eukaryotes there is another kind of ribosome. Eukaryotes कौन होता है? These are the cells where all the cytoplasmic organelles are present. Nucleus is demarcated by nuclear membrane. तो प्रोकैरियोटिक कहाँ खोया हुआ है? उस जो प्रोकैरियोट जो है, that means they do not have nuclear membrane and they do not have those cytoplasmic organelles bound by membrane. Means mitochondria, endoplasmic reticulum, Golgi bodies, lysosomes, they all are absent. In eukaryotic cells, they all will be present. And nucleus is bounded by nuclear membrane. Prokaryotes में nucleus होता है, there is nucleus, but without nuclear membrane. It is dispersed in the cytoplasm. Examples of prokaryotes are bacteria and blue-green algae. Not getting? Examples of prokaryotic cells are bacteria and blue-green algae, and all living organisms having eukaryotic cells. This is, कल भी ये लेकर पूछा था ना, this is fresh batch. We have started yesterday, and we have started with physical anthropology. So what is the problem? Did you attend class yesterday? Rishi Madirala, did you attend class yesterday? I think you did not. That's why. We have done in the first class basics of anthropology. Then we have started this batch with physical anthropology, and this is the basics of physical anthropology, and this is undoubtedly a fresh batch. So there are two types of ribosomes: prokaryotic ribosomes and eukaryotic ribosomes. And every ribosomes have two units. One is smaller subunit, and one is larger subunit. Five ways RNA. लिख लिया. If you cannot remember these 16S, 23S, simply write RNA and proteins. That will work. So every ribosome has two units. One is smaller subunit, other one is larger subunit. In case of 70S or prokaryotic ribosomes, there is 30S smaller subunit and 50S larger subunit. And you can see in 30S there is 16S rRNA and proteins. In 50S there are 23S rRNA, 5S rRNA and protein. Here you can see there are two subunits. One is smaller and one is larger. And you can see this. If you cannot remember, just simply write rRNA and protein. So, what are the functions of ribosome? So, ribosome जो है, it is a platform on which all the protein synthetic machineries are assembled. Like in a factory, all the machines are assembled and we produce some product. So here, ribosome is a factory site where all the protein synthetic machineries, means machineries required for protein synthesis, they are assembled on the platform of ribosomes. And it helps in the production of protein. That is why ribosomes are called protein factory. So there is a topic called protein synthesis. And in that topic you will see how different machineries are assembled and ribosome is helping produce protein. And second function is, while proteins are synthesized, it provides protection to newly synthesized proteins. जो दो subunits we have seen, they can be associated and dissociated. If you increase the magnesium concentration, both subunits associate. If you decrease the magnesium concentration, they will be dissociated. So proteins are synthesized between these two subunits. So it will be this length of protein will be covered by these subunits. So the part of proteins which are covered by these subunits, that gets protected from the action of various enzymes. इसके बीच में से जाते हैं, ये पार्ट कवर्ड रहेगा ना, दोनों के बीच में ऐसे मान लो जा रहा है, ऐसे कवर्ड है. The part which is covered by these two subunits, that part get protected from the action of enzymes. So that's why when proteins are synthesized, they do not get degraded. So that is the secondary function. Provides protection to newly synthesized protein. ठीक है. Now come to centrosome. So RNA से पहले क्या लिखा है? rRNA, ribosomal RNA, उससे पहले लिखा ACE. ACE stand for Sedvar unit. सेदवार्ग यूनिट. हर चीज का मेजरिंग की यूनिट होती है. राइबोसोम्स की जो आरएनए प्रोटीन दे आर मेजर्ड बाय सेदवार्ग. अब आ जाओ सेंट्रोसोम. Centrosome is a star-shaped structure just above nucleus. You see the diagram of whole cell. देखा? Centrosome. It is made up of a protein called tubulin. Tubulin. ऐसे न्यूक्लियस था, ऐसे बनाया था ना, दैट स्ट्रक्चर इज कॉल्ड सेंट्रोसोम. इतना ही है. अरे सेल मेम्ब्रेन के बाहर कुछ सेल का पार्ट कैसे होगा? इट इज बिटवीन सेल मेम्ब्रेन एंड न्यूक्लियर मेम्ब्रेन. बिना सेल का, बिना प्लाज्मा मेम्ब्रेन का सेल हो सकता? Without membrane, nucleus can be present, as found in prokaryotes. But without membrane, how cell can exist? Then there will be no distinctive environment for cell. So this structure is called centrosome. What is the function of centrosome? अरे जीएस में भी काम आएगा. हाँ, जीएस में कोई तो बायोलॉजिकल साइंस कराता नहीं है. पूछा नहीं जाता. अगर पूछ लेगा तो क्या करोगे तुम? तब बोलेगा अब ट्रेन में चेंज हो गया, इसी बोल के एक साल फिर खर्चे करेगा. और देखो ज्यादातर लोग प्रीलिम्स इसलिए फेल करता है, वो लोग सारे चीज पढ़ते नहीं है, पूछता नहीं है. यूपीएससी माहिर है पूछने में से. दो प्रिंसिपल फंडा है प्रीलिम्स निकालने के लिए. एक तो सिलेबस जो भी है प्रीलिम्स का, जो चीज मेंशन है सब पढ़ो. और while you are studying, refer to at least 5 previous years preliminary questions. बहुत इंपॉर्टेंट है. क्वेश्चन क्या है? It will tell you trend. It will say pattern of question, and it will also say to you which questions are relevant for your exam, which are irrelevant. Because being a beginner, when you are going through history books, geography books, every fact seems to be important, but they are not. So who is going to say these facts are important or these facts are not? Previous year's question. I think these are the best guide to every student. लोग से पढ़ लेता है, क्वेश्चन देखता नहीं है. It will be better if you go to 10 previous years question और revise 3 to 4 times at least. ये दो काम अगर कर सकता है, प्रीलिम्स क्यों नहीं निकलेगा? गोलाई तो मानना होता वहां पे. लोग देखो पढ़ लेता है, वो रिवाइज नहीं करता. लोग पढ़ता है, क्वेश्चन प्रीवियस ईयर क्वेश्चंस को फॉलो नहीं करता. अरे यू पीपल आर वेरी मच लकी. आज की डेट में यू कैन गेट सेग्रीगेटेड क्वेश्चंस फॉर एवरी सब्जेक्ट. सेग्रीगेटेड क्वेश्चन. वही देखो, हम लोग क्या करते थे, एक फ्लैट क्वेश्चन खरीदता था, और जब कोई हिस्ट्री पढ़ने जाते थे, मैं जब आंसर नहीं पढ़ता था तो उसकी क्वेश्चन में पहले सेग्रीगेट खुद करता था. फिर क्वेश्चन देख लिया, फिर पढ़ा. मुझे कभी महसूस ही नहीं हुआ जब मुझे प्रीलिम्स के लिए कोई कोचिंग की जरूरत है. हाँ, व्हेन यू आर सिलेक्टिंग सम फैक्ट्स फॉर प्रीलिम्स, यू मे मिस सम फैक्ट्स, मे मिस सम क्वेश्चंस, बट यू नीड ओनली 100 मार्क्स ना. प्रीलिम्स इज qualifying only. It is not scoring that your extra marks will be added to final selection. और जो 200 का होता है ना प्रीलिम्स, 200 पाने की टारगेट करता ना, उसकी कभी प्रीलिम्स नहीं होता. हमेशा उतना ही वेटेज देना चाहिए जितनी जरूरत है. आपको 125 तक टारगेट रखना चाहिए. मुझे 125 लाना है किसी से, क्योंकि अगर मान लो कंपटीशन थोड़ा आसान हो गया, 125 तक ही जाएगा, उससे ऊपर नहीं जाएगा. तो दो चीज, व्हाइल यू आर स्टडीइंग बुक्स, रेफर टू एटलीस्ट 5 प्रीवियस ईयर्स क्वेश्चन. अच्छा होगा अगर 10 साल तक चला जाए, तब कोई प्रॉब्लम ही नहीं होगा. और रिवाइज करना चाहिए, क्योंकि जो प्रीलिम्स में डायरेक्ट आंसर जो होता है ना, मैक्सिमम 15-20 ही लोग कर पाता है, बाकी सब एलिमिनेट करना पड़ता है. अगर आपके पास अच्छा रिटेंशन है, अच्छा अंडरस्टैंडिंग है, तो काफी क्वेश्चन श्योरली एलिमिनेट कर देगा. तो ये जो नहीं करता है, जो सिलेक्टिव स्टडी करता है, रिवीजन देता नहीं है, वो लोग पीटी फेल होता है. यू नो द पर्पज ऑफ प्रीलिम्स, व्हाई प्रीलिम्स conducted by UPSC? To screen out non-serious candidate. क्योंकि if UPSC is going to check lakhs of copies, it will be very difficult for UPSC to evaluate qualitatively. तो क्या है, all students are not serious. Those are non-serious, they should be smoked out. और जो पीटी पास कर लेता, it is believed that they are somehow serious. चलो इनका पेपर लिया जाए. तो आप लोग सीरियस बना दिए उसको. वही कोचिंग पे डिपेंडेंट रहना है. कोचिंग वाले जो करा रहे उसी को पढ़ के जाना है. कोचिंग वाले कोई भगवान थोड़ी ना है. इसके लिए कोचिंग की कोई जरूरत नहीं प्रीलिम्स के लिए. प्रीवियस ईयर्स क्वेश्चंस आर द बेस्ट गाइड. सच बताओ कितने क्वेश्चन देखे हो अभी तक? कितना क्वेश्चन देखा? तो जब भी कुछ भी पढ़ने जा रहा है, क्वेश्चन पहले पढ़ो. ऐसे पढ़ना नहीं, उसको अंडरस्टैंड करना. तब फेल ही नहीं करेगा कोई प्रीलिम्स. अरे आजकल तो प्रीलिम्स सस्ता हो गया, कोई ऑप्शनल है ही नहीं. अब आ जाओ न्यूक्लियस. फंक्शन, अच्छा अच्छा, फंक्शन जो है, इट हेल्प्स इन सेल डिवीजन. देखो, when cells divide, then chromosomes get separated. Suppose this is a dividing cell. centrosome है. These are spindle fibers. These are called spindle fibers. And these are called centrosome. Okay. So this is one centrosome, this is another centrosome. So these spindle fibers, they pull and push chromosome towards poles. So these chromosomes are pulled towards, this is pole, pushed and pulled by these spindle fibers. And these spindle fibers arise from centrosome. Can you see, Raj? These are the spindle fibers. This is spindle fiber, this is spindle fiber, this is spindle fibers. So these spindle fibers are arising from centrosome. They pull as well as push chromosome towards poles. And these spindle fibers are arising from centrosome. So that's why I have written it helps in cell division. Why we said 70 years in prokaryotes? Because 50 plus 30 is 70. अच्छा सवाल पूछा निंगशन. तुम लोग पूछे नहीं, कुछ पूछता नहीं है. अरे मैंने वहां पे एक का 30 दिया. आई हैव गिवन 30 यूनिट टू वन सब यूनिट, 50 यूनिट टू अनदर सब यूनिट, एंड देन आई हैव रिटन 70 years and you didn't ask anything. उसमें लिखा 40 years, then इसमें 40 years, then 60 years, and I have shown 80 years. क्वेश्चन तो बनता है ना. निंगशन वही पूछ रहा है. निंगशन, I should, whole class should thank to you, because when two subunits adhere, or associates. In that process, 10 Sedberg birth unit is lost. That's why it is, instead of becoming 80S, it becomes 70S. So while these two subunits are joining or adhering, it loses 20 S Sedberg unit. That's why it becomes 80S. तो निक्सन got it. अभी नहीं है अनुपम, कल आया था, कोई जानकारी में हुआ ये फिलिप्स कितने लोगों का हुआ लिया इसमें? नहीं यूपीएससी तो दिया तो होगा ना, इतने लोग क्वालिफाई किया। अच्छा, वैकेंसी 700 ना 800 बढ़ ही जाएगा, 800 तो ही जाएगा, 800 प्लस ही हो जाएगा वैकेंसी, फाइनल वैकेंसी जो है। चलो कम नहीं है, बहुत है, ठीक है। तो सेंट्रोसोम हो गया, अब न्यूक्लियस आते हैं। So nucleus, I should repeat the diagram. So this is actually problem क्या ना, इसमें काटो तो ऐसे होता है। Nuclear membrane is not a continuous membrane. It is interrupted by nuclear pores. समझे ना? So these are not wide open. These pores are fitted with nuclear annuli. ऐसे not wide open channel. There will be some structure. If it is wide open pore, then anything can come out, anything can go out. So this membrane is a double membrane structure. This is outer membrane, this is inner membrane. This is intermembranous space. And this is nuclear pore. The size of nuclear pore is 80 nanometer. 80 nanometer means molecules having size up to 80 nanometer, they can cross through nuclear pores. Molecules having size beyond 80 nanometer, they cannot pass through nuclear pores. And the structure of nuclear membrane resembles the structure of plasma membrane. Same structure exists. ठीक है। जब सेल डिवीजन करेगा तब अंडरस्टैंड कर लेगा सुबह से। अभी तुम्हारा अंडरस्टैंडिंग की जरूरत है, दैट इज functions of centrosome. And we have a topic called cell division. Okay. So structure is like structure of plasma membrane. So what is the function of nuclear membrane? It selectively allows molecules and ions to enter and exit through nuclear membrane. So it also doesn't allow every molecule to enter and every molecule to exit. If molecules are having the size up to 80 nanometer, then only they can cross through. And some electrical gradients are also determining entry and exit. So, like plasma membrane, it is responsible for selective permeability of molecules and ions across the nuclear membrane. समझा? So this is nuclear membrane. This part is called nucleolus. This condensed pink part. And this is called nucleoplasm. Also called chromatin. Containing DNA and protein. The nucleoplasm is also known as chromatin, containing DNA and protein. So I will draw structure of DNA, then you can see the structure of chromatin. I am going to discuss chromatin right now after this topic. So you can see the structure there. Now see the functions. Functions of chromatin or functions of nucleoplasm is heredity. It is DNA which pass on characteristics from one generation to another generation. समझा ना? The characteristics we inherit from our ancestors. It is with the help of DNA, deoxyribonucleic acid. And what is nucleolus? Actually, this is a part of nucleus where specific part of chromosomes, specific part of all 46 chromosomes come together, and it appears a rounded body under electronic microscope. Every 46 chromosome has a specific part. Not every 40. Out of 46 chromosomes, some chromosomes have specific part which is called secondary organization. समझा ना? When chromosomes having this secondary organization or secondary constriction come together, it gives an appearance of ball-like structure. After this topic, I will discuss chromosome. Then you will see what is the specific part. So that specific part in some chromosomes are called secondary constriction. When chromosomes having secondary constrictions come at a point, then it gives a ball-like structure under electronic microscope. Look, in a human cell, how many chromosomes are there? 46. Out of 46 chromosomes, some chromosomes have secondary constriction. Not all the chromosomes. यहाँ तक clear है? So there are some situations when chromosomes having secondary constrictions, they come together. And now, if you take a photograph with the help of electronic microscope, it appears to be a ball-like structure. That structure is called nucleolus. So in some phase of cell cycle, cell goes through a cycle. It is not a static entity. So in some phase of cell cycle, there will be nucleolus. Sometimes they disappear. Again they reappear. क्योंकि वो पार्ट अलग अलग हो जाता है तो डिसअपियर हो जाते हैं. अगर एक साथ आ जाता है फिर रीअपियर हो जाते हैं. वो सब डिस्कस करेंगे. पहले वो पार्ट पहचानने आना चाहिए ना. नेक्स्ट टॉपिक में मैं क्रोमोसोम करूंगा. सो व्हाट इज द फंक्शन ऑफ न्यूक्लियोलस? सी, दिस part of nucleus is responsible for the synthesis of rRNA. ये जो rRNA देखा है ना राइबोसोम में, these rRNAs are synthesized by nucleolus. That means indirectly they are responsible for the formation of ribosome. So nucleolus is responsible for the synthesis of rRNA, and you have seen major constituents of ribosomes are rRNA. That means indirectly it is responsible for the formation of ribosome. कितनी टेंशन में आ गया क्या? ऐसे लग रहा है. देखो, जो फिजिकल है इट इज लाइक अदर पार्ट. मैंने किस पैटर्न पे बात कर रहा हूं? सपोज आई हैव टेकन द पार्ट ऑफ न्यूक्लियस. I have shown components of nucleus. Then I have talked about different components. In nuclear membrane, I have shown the structure. Then I have talked about structure. It is resembling the structure of plasma membrane. Then I have talked about functions. तो यही तो याद रखना है. और टेक्निकलिटीज यू डू नॉट रिक्वायर. जो जैसे कि स्ट्रक्चर ऑफ एन्यूली, उसका रिंग जैसे साइकिल का स्पोक होता है ना, व्हील्स ऑफ साइकिल, जैसे स्पोक इस तरह का ही बना होता है. सो आई एम नॉट डिस्कसिंग एंड आई विल नॉट डिस्कस विथ यू. इट इज नॉट रिक्वायर्ड इन एंथ्रोपोलॉजी. लेकिन अगर जूलॉजी होता तो मैं इसको डायग्राम जरूर बना देता. यहाँ पे मैं क्यों बनाने जाऊँ? आई एम नॉट entering deeply into this topic because you don't require it. इतना तो कर ही सकता. And it is a new subject. There are some technical terms. You have to remember it. Otherwise things are not very difficult. क्या है centrosome में? क्या बोला मैंने? It is made up of a protein called tubulin and gives a star-like figure.